Hi friends, today we are going to see about hepatitis B, and the video on hepatitis A, it is on the description. The link is on the description and you can, don't wait just a moment, we can go to the lesson. Okay, and what is hepatitis? So already I have told you, hepa means liver and itis means inflammation. So the inflammation occurred in the liver is known as hepatitis, and there are 5 types, more types is there. Okay, now we are going to see about hepatitis B. Before we are going to the morphology and pathophysiology as well as clinical features, treatment and prevention, we are going to see about the genotype. Okay, normally the genotype here is 8 genotype. Okay, what are there means? They are A, B, C, D, E, F, G and H. So these are the 8 genotypes which is present in hepatitis B. Okay, coming to morphology, the shape of the virus will be spherical. And the envelope will be Australian antigen, or it is also known as HBS antigen. HBS means hepatitis B surface antigen. Before that, we have to know hepatitis B is also known as Dan particle or Australian antigen. Okay, and what are the structures here presenting in this board is, first, this is the hepatitis B surface antigen. Okay, hepatitis B surface antigen. Here the red one was hepatitis B core antigen, and the marking which is present here between the surface antigen, a thing which is present here is hepatitis B E antigen. Okay, and this is a polymerase, DNA polymerase, and this is a double stranded DNA. So this is what the structure which is present here, and genome will be double strand DNA. Capsid is icosahedral capsid. Because it is equilateral triangle like structure, the capsid is. So icosahedral capsid. Okay. Now let us see about the pathogenesis. The disease named as hepatitis B, or it is also known as homologous serum jaundice, and the reservoir in this disease will be human and no animals will be the reservoir, only the humans will be the reservoir. And source, it is caused by blood and blood products. And the mode of transmission is divided into 2 things. One will be direct and another thing will be indirect. Direct is further classified into direct, which means direct ingestion of any scalpels, scissors, okay, and endoscopes. So due to this ingestion, the virus will be entered. The virus will transmit from outside to the inside of the body. Direct. And the next one will be inoculation. Inoculation of any syringes, injections, needles, and when we are tattooing also, the virus can be transmitted. And the next one will be perinatal. Perinatal will be further classified into antenatal, intranatal and postnatal. Antenatal means the transplacental, okay, when we are in the womb of the mother, so the virus will be spreading through the placenta, okay. So transplacental will be antenatal, and intranatal during the birth. It can be, the virus can be transmitted, or postnatal, the infection will be spread during the feeding of breastfeeding, okay, feeding of milk, okay. Now we have to see what is the pathophysiology, what the virus do to our body, how it enters the cell membrane and the nucleus, okay. So first of all, normally hepatitis B virus is classified into acute and chronic. So as I told, in the hepatitis A, it will move to the virus will be transmitted and spread through the blood, and it will reaches the liver, okay. So 90% of the cases will be completely recovery in the acute hepatitis, but in case of chronic stage, in case of chronic hepatitis, what happens? So hepatitis means liver. So in the liver there will be a cells known as hepatocytes. See this, if this is a cells, I will take one cell, okay, I will take a single cell. So it has a receptor. Normally all the cells and all the membrane has the receptor like this. So when hepatitis B virus, let us assume this is the hepatitis or vigorous hepatitis, okay. Okay, and this hepatitis, it binds to the receptor, okay. The hepatitis which enters into the body, it reaches the liver, it reaches the hepatocyte of the liver, and in the hepatocyte there are more receptors, okay. So it binds to the hepatitis B virus binds to the receptor in the hepatocyte of the liver, okay. After reaching the hepatocyte, see, normally our cell membrane, I will separate the cell membrane now. See, this is a cell membrane, this is our cell, okay. And here this, see, this is a let us tell, yeah, this is the virus, okay. This virus enters the cell membrane. This virus enters the cell membrane. Let us assume the virus here. Double stranded and DNA polymerase is present here. Any doubts? Okay. So this structure will enter through the nucleus. How it is entering the nucleus is, see, this is a nuclear membrane. So this is how the nuclear membrane is present through the microtubules. We know cell has microtubules. So through the microtubules it enters. How it enters? Without the core. So this is the core which is present, but without the core it enters. So this is a double-stranded DNA, and this will be the polymerase. Only this part will be released from the cell membrane to the nucleus. And here what happens? They are collide now, okay. They are collided. The double stranded should be made as a single strand in the nucleus, or coiled or super coiled, okay. So what happens now? This double strand DNA will form like this. By some proteins and enzymes which is present in the nucleus, it forms into a single layer coil, okay. and the DNA polymerase also will be present in here. So now what this DNA polymerase does, it will produce mRNA. Okay, mRNA. So from the first I will tell. So there is a liver in acute stage. Completely 90% of recovery will be there in the acute stage. But what happens in the chronic stages? The hepatitis B virus which is present, it will bind to the receptor of the hepatocyte which is present in the liver, and it will beyond it will goes on entering the all the parts, and it reaches at last, it reaches the cell. In the cell membrane, after reaching the cell membrane, it wants to reach the nucleus. But before reaching the nucleus, there is a microtubule. Through the microtubule, the viral particle, the core, the DNA strand and the polymerase will get entered from the extracellular to the intracellular. Okay, and from the intracellular, the core will be getting removed. Okay, the core will remove. Only the DNA strand and the polymerase will be entering the nucleus. After entering, through any proteins and enzymes which is present in the intracellular, so it get coiled. Okay, the DNA strand become coiled, and the polymerase will be attached there. And this mRNA, this polymerase will produce the mRNA, and this mRNA with few more steps, it reaches and it produces a malignant, and the cancer cells will be produced and spreads to all parts of the body, which leading to invasive cancer. Okay, so this is what the pathophysiology. Okay, now we are going to lab diagnosis. So before lab diagnosis, what are the symptoms which is present? In acute stage, same as of hepatitis A, fever, malaise, arthralgia, urticaria, rashes will be there, and glomerulonephritis will be present in the acute stage, which can be recovered completely. But in chronic stages, the cancer is formed, and hence the liver become shrunken. Okay, the shrunken liver is known as cirrhosis. So chronic stage has the symptoms of cirrhosis, as well as it has a HCC, hepatocarcinoma, hepatocell carcinoma. So it has both things, main one cirrhosis, shrunken of liver, and HCC, hepatocell carcinoma. Okay, so these were the symptoms which is present here. And coming to lab diagnosis, the specimen which was used and collected from the patient is blood products will be there. Blood products and serum can be collected. Okay, so microscopy, we have to check this. Through the microscope, we have to check the shape is spherical or not. Envelope, hepatitis B surface antigen will be present, and genome double stranded DNA, capsid, icosahedral capsid. So this can be observed through the microscope. And coming to cultural characteristics, we have animal culture and human culture. Okay, so in vitro, in the lab the culture can be done. And serological test, so serological test are main. So this hepatitis B surface antigen and anti-hepatitis B surface. So here in this cases, this antigen will be appear at the initial of after the infection, the surface antigen will be appear in the blood in our body, and it will appear in the initial infection. After it will goes to the peak, it reaches the peak in 8th to 12th week. Okay, and it will disappear in 24th month. So this is what the thing. And here hepatitis B E antigen and anti-hepatitis E antigen also will be present in the body, hepatitis B C antigen. So this, it will appear in the body after 1 week, after first week. Okay, so this is about the serological test. And the normal test which is present here is ELISA, enzyme linked immune sorbent assay, can be done. And any other test like immunochromatography test and lattice particle agglutination test. Lattice particle agglutination test. Okay, other test or like alanine aminoferous test and bilirubin test can be done. Molecular method PCR can be done, polymerase chain reaction. And HPV DNA test, hepatitis B virus DNA test can be done. Okay, and one more thing I have remember that incubation period. Okay, incubation period will be 1 to 6 months. For HPA, hepatitis B, hepatitis virus A, 2 to 6 weeks. Hepatitis virus B, the incubation period will be 1 to 6 months. And portal of entry, it will enter by skin and mucosa because it is transmitted by blood and blood products. Mainly blood will be transmitted through what? The skin and mucosa membrane. Okay, so skin and mucosa will be the portal of entry, and the site is mainly liver. Okay, and the next one will be prevention. Okay, so same as hepatitis A, it can be prevented by 2 ways. One is active and the another one will be passive. Active immunization can be done by plasmid vaccine, cloned vaccine and synthetic peptide vaccine. Plasmid vaccine, cloned vaccine and synthetic peptide vaccine. Plasmid derived vaccine. So plasmid derived vaccine, cloned vaccine and synthetic peptide vaccine. In passive stage, passively we can give 0.05 to 0.07 normal pooled immunoglobulin. Okay, milliliter per kg normal pooled immunoglobulin can be given for 48 hours. Remember that. Okay, and treatment, so treatment is not yet available, but we can give interferon alpha. Okay, so interferon alpha can be given. And in the chronic stage, for acute we can give interferon alpha, and for chronic we can use lamivudine with lamivudine, interferon alpha plus lamivudine, and also we can give famciclovir. Famciclovir can be given. Okay, so this is about the hepatitis. Treatment will be interferon alpha for the acute hepatitis, and for chronic hepatitis we can give lamivudine, famciclovir. Okay, so this is about hepatitis B virus, and at the next video I will be teaching about hepatitis C virus. Until then,